Good evening to the invited guest of honor, Professor A.M. Deshmukh sir, our chief guest, Dr. Swapna Mukherjee madam, resource person, Dr. Gopalji Gopal sir, and dear participants. I am delighted to offer a gracious welcome to all of you in this valedictory session. The fragrance of flowers is spread only in the direction of wind, but the goodness and knowledge of persons spreads in all directions. So we are delighted to have with Dr. Swapna Mukherjee Madam as our honorable chief guest of the valedictory session. Madam is head and associate professor in Kolkata, University of Kolkata, Dinbandhu Andrews College. She is also general secretary and West Bengal state president of Microbiologist Society, India. Her research interests are transgenics and natural product chemistry, environmental microbiology.
So I request participants to share his or her feedback quickly.
Sir, myself,
Welcome,
Dr.
welcome.
Megha Kulkarni.
Yes.
I
Welcome,
participate.
madam.
Yes, sir. Sir, namaskar, Deshmukh sir.
Namaskar.
My
namaskar.
greetings to Dr. A.M. Deshmukh sir
Yes, thank you.
and Dr. Sapna madam. Sir, I am very much delighted to attend this webinar as it was very informative and the discussions during the webinar were very fruitful for us to gain more knowledge because nowadays the techniques are modifying and developing and because for the research purpose I found them very fruitful because I'm from the old batch and I just wanted to gain more knowledge in the field of rDNA technology. And uh, on that, taking that purpose in my mind, I joined the webinar, and I enjoyed the webinar very much, sir. And today I am very much delighted to say that everybody should join the webinar so that the group discussions, which is a skill, will help to develop during such interactions. So thank you, Dr. Gopal sir, Dr. Deshmukh sir, Dr. Sapna madam. Thank you.
Thank you, madam. Thank you. You have not introduced, madam. Hello. Yes.
Sir, myself, I'm working as a HOD, Department of Microbiology, Chintamani College of Science, Coburna. And also I'm working as a coordinator for Microbiology Society India on the behalf of the Gondwana University, Gadchiroli. I'm POS member over Gondwana University, Gadchiroli, and I have got a guideship for the microbiology subject from the Gondwana University, sir.
Great, great. Thank you, madam. I also feel proud that you have attended regularly. I watched you have attended regularly. I know you are
Yeah,
teaching,
yeah.
but I was not knowing that
Only
you are HOD and you are
no, sir.
microbiologist representative.
Actually, I am very happy to join the course, sir.
Thank you, madam.
Thank you, sir.
Any other
Very
participants?
good. You have sincerely attended the course.
Yes,
Yes,
sir.
yes,
Sir.
madam has attended course. When
Sir.
she could not attend, then she send message me. कि आज
Yeah,
नहीं
sir.
attend कर पाई मैं.
Only thing, sir, myself and my daughter, we
Yes.
got the COVID infection. So during that 2-3 days, we could not attend properly.
Thank you, madam. Any other
But thanks,
participants?
sir.
Why you are hesitant? I don't understand. Participants, please share your view.
Any
Hello.
other participant?
Yes.
Yes.
Hello.
Yes.
Good
Tell
evening,
me.
sir.
Yes,
Good
madam.
evening.
Dr. Deshmukh, Dr. Swapna, and Dr. Bukranj, Gopal sir. I am Ruella D'Souza, and I am an assistant professor in St. Xavier's College, Goa. And our HOD suggested that this was an opportunity that had come up, and she said it is an opportunity that we can take you just grab it because a webinar, it's something about hands-on. I mean, in the sense that it is about techniques that we don't usually cover. So we decided, actually there are, we are 1, 2, 3, 4, 5 of us who are attending this course from St. Xavier's. And one thing I realized is that if you are colleagues or friends attending a course together, there is a discussion that happens. It so happens that because of COVID now we are also on a break, but we also, when something was happening and in case somebody was hesitant to ask, or later on we messaged each other and said, what did you think of this? Can we integrate this? So have you heard of this? And with that we have picked up lots of insights and pointers on what sir has been teaching us that has been very helpful. So it has been a nice learning experience. Us teachers getting back to the classroom and online class has been a revelation, and incredibly grateful. One thing I would like to say, Gopal sir has this technique to encourage the students to speak up. If nothing else, at least he asks you the question which makes you want to contribute, because when you contribute, even if it's a yes or a no, it leads the discussion further. So even if one student responds, the rest 50 of them are benefited because of one person's question. So Gopal sir, I appreciate and thank you very much for that. Thank you so much for this, and on behalf of all of us, I'd like to say great going. 17 batches of this course means there is something to it, and I'm very grateful that as the participant of the 17th batch, I have got a lot to gain from it. Thank you so much.
Respected Deshmukh sir, Swapna ma'am, and Gopalji sir, good evening. And
Good evening.
सर ये कोर्स मैंने सडनली एक फ्रेंड से पूछा था कि क्या अभी कोई इंटर्नशिप कहीं चल रहा है, तो उसने मुझे शेयर किया कि ये अभी चल रहा है। तो मैंने लास्ट ज्वाइन किया, 17 को स्टार्ट होने वाला था और मैंने 16 को, मतलब कि जबकि डेडलाइन खत्म हो गया था, तो गोपाल जी सर से मैंने कॉल किया, सर अभी टाइम है क्या, ज्वाइन कर सकता हूँ मैं? तो सर ने बोला कि अच्छा आप ज्वाइन कर लो। फिर मैं लास्ट डे ज्वाइन किया, तो फिर ज्वाइन करने पता चला कि नहीं, सच में ये कोर्स काफ़ी अच्छा है और मुझे बहुत ज़्यादा हेल्प करने वाला, क्योंकि मैं अभी MSc फर्स्ट ईयर में हूँ। और गोपाल जी सर का सबसे बेस्ट पार्ट ये है कि सर का जो एनर्जी है, मतलब जिस वे से पढ़ाते हैं, तो काफ़ी ज़्यादा एक्साइट करते हैं सर का, मतलब टीचिंग वे जो है। और सर का जो क्वेश्चनिंग है, आप गलत हो या सही हो, आपका सर का क्वेश्चन इस तरह से पुट करते हैं कि अंदर से जी होता है कि गलत हो या सही हो, काश सही हो तो ज़्यादा अच्छा है, लेकिन गलत भी बोलने के लिए मतलब एक्साइट करते हैं सर की, नहीं तुम बोलो जो भी तुम्हारे दिमाग में, ताकि कुछ अलग भी सोचने का मौका देते हैं। कई बार होता है कि हमारे जो टीचर्स होते हैं कि स्ट्रक्चर्ड आंसर चाहते हैं, लेकिन सर का ऐसा कि आप कुछ अलग भी दिमाग लगाओ तो ज्यादा बेटर है। ये चीज मुझे काफी ज्यादा अच्छा लगा। थैंक यू सर।
Yes, sir. I am student of MSc in Sant Gadge Baba Amravati University. I'm really glad that when our HOD told about this course and we joined this course, it was really informative for us, and the discussion that sir carries on, like he does not only teach, because some teachers what they do is they directly teach and do not do any discussion work, but sir carried out the discussion work, and that was really good for us. Whatever doubts were there or whatever ideas we had in our mind, we could easily put on that ideas and confirm from sir whether it is applicable or not. So that was really good.
सर आई वुड लाइक टू थैंक यू सिंसियरली। कल आपने मेरी बहुत बड़ी मदद कर दी, क्योंकि मैं जेन्युइनली PhD के लिए ट्राई कर रही थी, बट मैंने आज तक जितने भी SOPs भेजे मैंने विदाउट मेथड्स ही भेजे, एंड आई केप्ट वंडरिंग कि मेरा हो क्यों नहीं रहा था। बट कल आपने लिटरली वो जवाब दे के बहुत अच्छा मतलब गाइडेंस दिया। सो अब मैं विद मेथड्स ही SOP भेजूंगी मेरे, so that I would get selected.
I think I really enjoyed the session. I personally love genetics because of my teachers. I'm for my teacher and BSc, and I think sometimes we take it for granted that we know certain part, But there are certain things that we don't really just skipped our mind, and so I think those are those portions where you have touched upon. Like I took it for granted. Okay, you just we put the DNA into the plasmid, and that's it. But there are so many nitty-gritties that go into that, and I think you have touched upon that. So it has helped me grow a lot. And I like everybody else has said, I really appreciate the way you have encouraged the participants to speak up often, and then you waited patiently. So we also, as Ruella has said, we have been teachers, we are on the other side, and it's quite like you're waiting for students to speak up, and you have been very patient, sir, and you have made sure that somebody has spoken up, and that has carried on the discussion, as Ruella has just said. And I think what added more to it, we were all looking forward to at least some practical portion, but I think without actually having practicals, you have brought the practical component, because with uh, like actual examples of people having worked in the lab and what are the results and what we're looking forward to. I think that was something I have not seen, at least in neither in my MSc or during my BSc. So thank you for that, sir. And yeah, I really enjoyed, and it has taken me, given me more than what I already know. So I'm more in love with genetics. That's all I can say. So thank you so much, sir, for everything.
Very, very good reaction and very positive comments. But
Yes.
one thing again, you are not introduce yourself from which
So
institute
sorry,
you are.
yeah, I'm Dr. Valerie Gonsalves. I'm assistant professor at St. Xavier's College, Goa.
Good evening, everyone. I'm Pratik, and I'm currently in the final year of my BTech Biotechnology from Dr. D.Y. Patil, Bioinformatics and Biotechnology Institute, Pune. So, about this course, I think in my third or fourth semester in BTech, RDNA technology was taught by our professor. But in colleges, what happens is the professors have the aim to complete the subject very quickly, and they want to complete it just for the sake of completion. But here I learned many new things about our DNA. Not only, of course, my basics were clear, but here more practical aspects were discussed. Gopal sir were more interactive with the students, and every questions asked he would answer really well. He was well prepared as well, and other two faculties as well, Dr. Ravi and Dr. Abhishek sir. Both were really prepared uh, before their this lectures. And one thing I think the course could be uh, been more effective if instead of 3 continuous hours, the course could have been reduced to a limited number of hours each day, and the days could have been increased. Just because sitting here for 3 hours and constantly looking at computer screen is sometimes really difficult, and the concentration keeps weathering off. So yeah, that's only my suggestion. And overall, I think the course was really effective and fruitful. Thank you.
Thank you. Thank you for suggestion. Thank you. Okay.
Good suggestion given. Definitely, we will discuss among ourselves
Yes,
and
yes,
we will
yes,
see what can be done.
yes. Any other participants? No. अभी तो बोल लो,
Okay.
मेरा सुनते रहे 11 दिन तक. अभी तो बोल लो भाई. Okay. So now, Tiwari sir, if no one is willing to share, then continue, please. Arifa, Himanshu, no one. कुछ लोग तो सर काशी. Hello. Someone is just, yes, forum. Okay. Some participants are from Kashmir also, but their internet is fluctuating. So
Yeah, that is a problem in Kashmir.
yes, yes, yes. Sometimes they are facing problem. Anyway, that's fine. Please continue, sir. Vipran sir.
I'd like to request Madam to share now few words with her. Please, Madam.
Yes. Yes, madam.
Okay, good evening, everyone, and thank you, everyone. Respected Professor Deshmukh sir, Dr. Gopalji Gopal sir, Dr. Biprangshu Kumar Tiwari sir, other dignitaries and participants. I'm really feeling honored to share some quick thoughts with you about this course in this valedictory session. But before that, on behalf of Microbiologist Society, I would like to express my sincere appreciation and gratitude to all the coordinators and the participants for their very, very active participation, which I think have made this course very, very fruitful. What I get from Megha ma'am, Ruella ma'am, Iftikhar, Rukaiya, that all of you had 10 to 12 days of very, very productive discussions and interactive sessions, right? Given that there are very talented resource persons, what I see, to help participants accomplish their learning goals. To be very honest, I'm not actively involved with this course, but I have been so much impressed by the eagerness among the participants for this course organized by our society. Participants, you are the 17th batch of this course. When any course runs successfully for this long period of time, there is no doubt that the course content is very relevant and advanced. Actually, the topics included in recombinant DNA biotechnology in this course, what I've seen that they have been structured and sequenced very nicely and build on one another in a way that it allows students to integrate each new idea, topic, or theme with the preceding ones as the course proceeds. So I'm not going to summarize this coherent and very, very dynamic course. I'm just going to look ahead and want to mention some of the points. Participants, you must have learned that this RDT or recombinant DNA technology, this has the ability to directly manipulate the genes of organisms to create new proteins, right? Beside this technology, there is another technology developed in recent years and being widely adopted in research labs around the world is a CRISPR-Cas9 technology. You must be knowing that. This CRISPR-Cas9 genome editing system has become the poster child for innovation in synthetic biology these days and being used widely by scientists to edit the gene to treat lethal diseases like cancer. So it's so successful, and its success is in its simplicity and efficiency. But then also, this technique also has some limitations. And what I recently found in a report which was published in PNS in April 2021, some of the researchers at the Weiss Institute at Harvard University and Harvard Medical School, they have created a new genome editing tool called Retron Library Recombinaring, or in short, RLR. That can generate up to millions of mutations simultaneously and barcode mutant bacterial cells so that the entire pool can be screened at once. It can be used where CRISPR-Cas is not feasible or toxic, and obviously they are claiming that this results in better editing rates. So all of these subjects can be taken up in future. I see many of the participants are from different colleges and universities. They are professors, they are involved in teaching. So I'm requesting them to take up this one of these subjects to conduct various certificate courses, workshops or seminars, etc. in future days, which will be collaborated with Microbiology Society India. I must mention one more thing, that our society is conducting one more course on basics of genomics and proteomics. I think Dr. Gopalji Gopal has already mentioned that. Having said that, I cannot overemphasize the importance of such efforts in order to keep the momentum of the academic growth. I congratulate the whole team once again, especially Gopal sir. I see that he is very popular for the effort they have put in, for the kind of subjects they have addressed. And many of the participants have shared their views and they're given their reactions. And what I find that they are giving very positive feedback. They are very satisfied about the course. And at the end, I can say that I'm hoping, like everyone, that 2022 will be COVID fear-free year, so that more of this type of applied and activity-based courses can be conducted. Thank you all. Thank you very much.
Thank you, madam, for your influential message. We are very charmed by your sharp fluency and knowledge in the additive. Thank you very much, madam, for presenting. Now I'd like to request Professor A.M. Deshmukh sir. He is pioneer and pillar of the Microbiology Society. He is president of Microbiology Society India. He was former head and professor in Department of Microbiology, Baba Sahab Ambedkar, Marathwada University. So I'd like to request Deshmukh sir to share few words with us and encourage with his words. Please, sir.
Thank you. Thank you very much. Thank you, Dr. Tiwari, for giving me an opportunity to interact with the August gathering. Now I am using the word August gathering. Probably I would not have used before starting this workshop. But after the workshop, all you are so have updated knowledge. I can call you as the August gathering. Today, Dr. Swapna Mukherjee is here. She is a really hard worker, sincere, and working in Kolkata. She is the state president of microbiology for West Bengal. And the most important figure today is Dr. Gopalji and his two other research workers. It is a teamwork, of course, although Gopalji is a leader and his efforts, I want to appreciate his efforts. The reason is that Microbiology Society is running courses. Our first condition is that the fees should be affordable. And Gopalji has constantly kept the fees affordable to the student. Compare the hours he is spending to give the knowledge to you. It is near about 32 hours course. And for 32 hours course, we are charging only and only 600 rupees. And in the feedback also, I have observed that the participants, those who have attended sincerely, are very happy, very, very happy. And that is the beauty of the course. Students from different corners of India. What is our motto? Motto of Microbiology Society is Bio Make in India. Meaning is that Honorable Prime Minister is telling Make in India. Microbiology Society has selected one branch of Make in India, that is a Bio Make in India. And today, RDT, recombinant DNA technology, is indispensable in Bio Make in India. So, here every time in every batch, this is the 17th batch. I'm very happy because we are trying to educate the maximum learned people regarding RDT. And Dr. Gopalji and his team, they are explaining the subject in such a manner. They are giving the knowledge in such a manner that our graduates, our postgraduates, even PhD holders, they are becoming happy after attending the course. And that is the strength of the course. So, it's as a president of Microbiology Society, it is my duty to congratulate all you, including the students, because you are sincerely attending. I'm calling you as a student, although there are many teachers, because I myself also consider as a student of the microbiology. All we are student. Gopalji is also student because he is expert in RDT, but in biofertilizer he may be student. So every one of us is a student of microbiology, and we should try to learn many things. Now, lastly, I want to come to the microbiology society. I will request everyone go and visit our website www.microbiosociety.org. Very simple website. And we have a Telegram group for the student. We have WhatsApp group for the teachers. If you are not joined our any Telegram group or any WhatsApp group, please give me message on WhatsApp or give message to Gopalji on WhatsApp. We will add you, so that or permanently you will be associated with us. I will never say, because again you see, I never say that you should work for microbiology society. I'm always telling microbiology society is the bridge, is the way, is the road to achieve the goal. What is the goal? Goal is a microbiology. You love microbiology, you may not love microbiology society, but we have to change the India. Means completely we have to do the revolution in India in biotechnology. And Corona has proved that the time has come. Today, you cannot live without the knowledge of microorganism. In either microorganism may be injurious to the health, or microorganism may save our life. So we can handle the microorganism perfectly and properly. That's why, again, I request you, all the students, go visit the website, join the hands of Microbiology Society. Very recently, we have published the microbiology calendar, and we are freely distributing. You see, throughout the India, it is the first scientific calendar. Even after the freedom 1947, none of the subject has published a scientific calendar like microbiology calendar. And we are not making a business. We are distributing freely. Already we have distributed 1,500 calendars, and we have given order to more 1,500 calendars. Means the 3,000 calendars we are distributing free of cost. Why? Only to popularize the subject and only to create awareness. So today's attempt of Dr. Gopalji is to create the scientific awareness regarding the RDT. And I'm happy to report my reaction that he is successful in creating awareness regarding the RDT. Again, as a president of Microbiology Society, I wish all the best to his team, to all the students. I'm thankful to Dr. Swapna Mukherjee for attending this beautiful event and addressing the students. So I'm very happy, and say you goodbye for today. Thank you. Thank you very much.
Thank you so much, sir, for sharing your deep knowledge with us. Your words were really enthusiasm and valuable knowledge for us. And we are also very thankful to the Microbiology Society India, because this society always provides us platform to share our knowledge in microbiology field. So this is the moment of accomplishment and pride for our resource person, Dr. Gopalji Gopal. And this achievement also gives a sense of the responsibility. So I believe that Gopalji Gopal will continue this workshop in future also. And I request sir to say something about the programs in future.
Thank you, thank you. So, respected Deshmukh sir, Swapna madam, Viprans Tiwari sir, Megha madam, Ruela D'Souza. Now I come to know they are faculty, because I was considering that they are students. So, as I continuously trying to motivate participants. Lastly, also I can say that सूरज ना सही हम दीपक तो बन ही सकते हैं. If we will try to become a sun, सन ना बन सके दीपक तो बन ही सकते हैं. पूरे दुनिया को हम उजाला कर ना पाए, लेकिन हम अपने आसपास को तो उजाला कर ही सकते हैं. So we must, our intention must be to spread light, energy to other people. ऊर्जा का हमेशा ही संचारण करते रहना चाहिए, प्रकाशित करते रहना चाहिए दूसरे को. ये हमारा मोटो, हमारा इंटेंशन होना चाहिए. और मेरे पिताजी जो कहा करते थे वो आपसे कहता हूं, लगा दो जान मेहनत में, सफलता पांव चूमेगी, खुशी तुमको तो होगी ही, खुशी काफी मुझे होगी. So with this note, I again thank our guest, Swapna madam and Deshmukh sir and Viprans sir. Thank you very much.